So hello everyone, I'm very happy to be here with you to speak about this interesting program about cancer immunotherapy and the change in paradigms in cancer immunotherapy. So what we're going to do in this program is first of all give you an overview that I will give you in 30 minutes, and then you will get a series of different interesting talks about predictive biomarkers and so on that you will get in the subsequent part of this program here. So what I'm going to start with is to give you an overview of the cancer immunotherapy that we are speaking now. I'm Dr. Escudier from Gustave Roussy in the genitourinary group in my institution, and I've been involved in immunotherapy for many years now. So this is my disclosure. So just to go back to what we do in cancer, we have done a lot of different approaches to try to improve the cancer we see in our patients here. Of course, the first step has been surgery, and if you go to old days, the first surgery for cancer was done in 1846. Then the introduction of radiotherapy came at the early 90s, and in 1901 came the first machine on radiotherapy here. And then the concept of chemotherapy came in 1950, and of course you know that we developed a lot of chemotherapy, which of course is not targeted to something special, but try to stop the growth of tumor here. And recently, in the past 20 years, I would say, two new concepts arise and came to our new therapy for cancer here. So the first one is what we call targeted therapy. So the principle of targeted therapy is to try to find a target in some tumor cells and target these specific targets to try to stop the growth of the tumor. As an example, we worked on HER2 for breast cancer, on c-KIT for GIST, on VEGF for tumors who express VEGF. So that's what we call targeted therapy. The concept came in 1997. And of course, due to the discovery of a lot of biological new concepts, this targeted therapy became very popular at the beginning of the 2000th century, of course. So that's the first new concept which came here through ages. The second concept came not to target the cells, the tumor cells, but to target the host itself by trying to make the host more able to fight against cancer. And as we know, we have and we develop all over our life some abnormal cells that the immune system is able to get rid of. So the question is, why don't we get rid of all the tumor cells? And the question is that our immune system sometimes is not able to do that. And targeting the host and the immune system has been an idea for many years now. And if you go back to this old approach of immunotherapy, which we call the toxin from colase, that we use to try to develop an immune reaction against a tumor, it was used in 1899. So quite a long time ago, the idea that we could use the immune system to fight against cancer was developed here. And as you can see, if we inject the toxin in some tumor, we can, in sporadic cases, of course, unfortunately it's not in every case, we can see some decrease and some tumor shrinkage, which in these patients was very dramatic here. Does it go through PD-1 or PD-L1 interaction? We still don't know, of course, but that might be one of the ways this toxin has been acting here. And a lot of approaches about immune therapy has been done through the age. And of course, this is due to the fact that the immune system is made by different cells within the tumor environment. Some are good, some are bad. And you can see here the different players of the immune system. Some will bring the antigens to the immune system, which we'll call the presenting cells. Some will be good cells, which are activated T cells. Some will be bad guys. And of course, the tumor suppression and tumor progression will be a kind of balance. And getting a good balance in terms of tumor suppression, good guys, will make the immune system able to act against cancer cells here. So let's see about what can be done in a video which will give you a good idea about what is the immune system, how the immune system can work. And so you will see this nice video now.
main functions of our immune system are to monitor tissue homeostasis, protect against pathogens, and eliminate damaged cells. Research in recent years has shed light on the relationship between cancer and the immune system, and has positioned the immune system as a crucial regulator of cancer development. Cancer cells can avoid detection and destruction by immune cells. This can happen through making detection more difficult by reducing the expression of tumour antigens, expressing proteins that induce immune cell inactivation, and inducing cells in the microenvironment to release substances that suppress immune responses and promote tumour cell survival. While the immune system is important for recognising and eliminating tumour cells, it also plays a critical role in tumour progression. This dual role of suppression and promotion is termed immunoediting and consists of 3 phases: elimination, equilibrium, and escape. During elimination, most cancer cells are destroyed by the immune system. Certain tumour cells that survive immune destruction enter the equilibrium phase, which is characterised by immune resistance. The escape phase is the final step, where immunologically resistant tumours grow and establish an immunosuppressive microenvironment. As a result of this process, immunotherapies that stimulate the immune system or counteract signals produced by cancer cells that suppress immune responses have been highly sought after. For example, immune checkpoint modulators help stop immune checkpoint proteins from suppressing immune responses. This allows the immune system to become more functional, increasing its ability to destroy cancer cells. A second method is immune cell therapy. In one strategy, T cells that have infiltrated a tumour are collected, and those that show the greatest recognition of tumour cells are selected and grown. They are then activated by cytokines and reinfused to help shrink or destroy tumours. A third approach is antibodies. For example, antibody-drug conjugates link antibodies to a toxic substance that is delivered to a cancer cell when the antibody binds to it. Other antibodies induce apoptosis or recruit other immune cells to destroy the target cell. Ultimately, these immune-related methods continue to show promise in the medical management of cancer.
Of course, the anti-tumor effect is a dynamic process. It's not a single process where you have one cell acting against tumor cells. And you can see on this cartoon that we have different steps in terms of what happens when you go to the immune therapy system here. First thing that you have a relapse of cancer cell antigens, which is of course the first step when you have cancer death cells, where the antigen is released in the microenvironment. This antigen, of course, is not always known, but this antigen is supposed to be presented by what we call cancer antigen presenting cell. The main one being the dendritic cells, the antigen presenting cells here. Then based on this presentation to the immune system, we should have a priming and activation of the T cells. That's what we call the interaction between antigen presenting cells and T cells activation here. These T cells, which are activated now, should traffic and migrate to the tumor cells. And then in this microenvironment, this infiltration of T cells is going to have, obviously, if it works, an anti-tumor activity against the tumor cells here. And that's what we have in terms of killing the cancer cells by the immune system with this dynamic process, where we have first release antigens, presenting the antigen, then activation of the immune system, and then the T cell activated become killer cells, which are going to kill the cancer cells here. And that's the activation we try to get when we use immunotherapy with what we have done for many years now. So what we have learned in the past, I would say 10 years, is that we have some very important receptors. We call that the immune checkpoints, which are both on the part of the T cell, but also of the antigen-present T cells and on the tumor cells here. And these immune checkpoints have a very important role, both in terms of inhibitory or activating effect here. This is an example here of what we have between the T cells and the antigen-presented cells. The antigen here is presented by the APC here to a T cell receptor here. And then we have a second signal here, which becomes now a co-stimulation between CD28 and B7 here, which is an important part of the activation of the T lymphocytes here, and which is a required step for having this here. This activation is also then controlled by what we call inhibition of T lymphocytes. Why is it like that? Because the immune system is initially mostly done and built to fight against viral bacterial antigen here. So what has been done with the human system is that we, at a certain point, have some inhibition of the immune system just to avoid to have permanent activation of our immune system. And in addition to what we have activated signal, we have negative signal here, such as PD-1, PD-L1 interaction, which is one of the negative signal here, or CTLA-4, B7, which is another negative signal here. So these negative signals are going to make a break on the immune system. And that's the balance between activation and inhibition, which is going to explain what's going on with the immune system here. And of course, more we understand the immune system, more we understand the way it's activated and then inhibited, more we try to get a better way to use the immune system to fight against cancer here. So if you look now at what we have in terms of acquired immune resistance, which is of course something that we have in most of our cancer would develop cancer, patients would develop cancer here. We initially have innate immune resistance when we have PD-L1 and PD-1 which are expressed on the tumor cell and the T cell. So that's something which happens. In some tumor cell you have expression of PD-L1, in the T cell you can have expression of PD-1, and if you have both these expressions, then you have immediate, what we call innate immune resistance to immune system. So these are tumor cells which are going to grow whatever you do, except of course if you can break and stop this interaction here. You also have what we have adaptive immune resistance, which means that initially we have an active system and we have T cell activated by presenting the antigen to the immune system. But then, because of some pathway which are going to be activated by the T cells, like STATs and so on, you will have expression of PD-L1, secondary expression of PD-L1, which is going to be induced by the evolution of the cancer, which is going to stop the activation of the immune system and then to avoid that the immune system is going to get rid of the tumor cell. So two different mechanisms. One is innate, which means that it's from the beginning of the story. One is induced, which we call adaptive immune resistance, which is mostly coming with most of our cancer here. So this resistance mechanism to immune checkpoint inhibitor are of course something that we would like to try to get rid of. And this is what we call the cancer immunogram, which means that we have different ways we could act to try to re-stimulate the immune system and to try to fight against this mechanism of resistance to the immune checkpoint inhibitor. And this is different way to act against this problem of resistance, and of course to try to induce activation and effect of immune system here. And you have the different factors which are important in terms of how immune system is going to work here. One, of course, is the tumor load, and you understand that the antigens are very important. And the mutational load, so more you have antigens, more it seems that you can have immune system activation. And this mutation load is different from tumor to tumor, which might probably explain why some tumors are more immunogenic and some tumors are less immunogenic. So this is one player here. Of course, the lymphocyte count is important. If you don't have lymphocytes, you cannot stimulate your lymphocyte because you have not enough lymphocyte to act against tumor. The intratumoral infiltration of the T cell is also important. Not only you need to have lymphocyte, but you need to have lymphocyte within the tumor. These T cells within the tumor are going to get activated, what we call intratumoral T cells, and they are very important here. Then you have the PD-L1 as the other checkpoint inhibitor. If you can get rid of PD-L1 or if you can block PD-L1, then you might reactivate the immune system. You also have some very important inhibitors, which are, like an example, interleukin-6, C-reactive protein or others, which are inhibitors of the immune system here. If you can remove IL-6, CRP, and so on, TNF-alpha, and so on, from the microenvironment, you can also reactivate the immune system here. And then you have some other probably important factors, which are, like an example, glucose activation, and you need glucose in the tumor cells to grow the tumor. So if you can decrease the glucose level in the tumor, etc., this is also some important here. And finally, if we go back to the first part of the immune system, presentation to the cells is important, as I told you before. So MHC expression, which are important factors to be able to express and present the antigens to the immune system, is important, as well as interferon gamma, which is one of the important cytokines, which is part of the immune system activation I will see later on here. So let's go now to the general concept of PD-1, PD-L1, which of course has been the first important cause signal to be activated in cancer cells here. So this is, as I told you before, a very schematic presentation of what's going on with the immune system here. First signal, antigen recognition. Of course, you need to have an antigen, but this antigen is presented to the T cell, and you have the first step here. And then you need the co-stimulation, which is signal 2, which are important one to be part of the immune system part. And some of these co-signals, signal 2, is positive. Some of these co-signals are negative. And many of these negative second signals promote tolerance to the immune system here. Of course, co-stimulation is key here, but you have to know that some are positive and some are negative here. So this is the first step. You can see positive signal with antigen recognition here. Then, of course, you have another positive signal here, but you have a negative signal here when you have negative co-signal, which is going to make a break on T cell. As an example, PD-1, PD-L1, which is a negative co-signal here. And this is some of the co-signal which have been described in the past, I would say 10 years here. PD-1, PD-L1 is now more than 10 years knowledge, but we have some new concept coming on, and you can see that some of them are probably not yet described, but we have inhibitory and positive and stimulatory co-signal here. So that's certainly PD-1, PD-L1, one of the most important one, because we know exactly what's going on here, and also we know that PD-L1 expression on the tumor cells is probably one very important prognostic and predictive signal for activity of this interaction between PD-1 and PD-L1 here. So that's something that you have to know. CTLA-4 is also very important here, which is interesting between the B7 is both activated by CD28 and inhibited by CTLA-4. So that's a very important signal here, co-signal, which can be on the activated or inhibitory side here. So let's go back to this cartoon here. So what do we get when we have activation of the immune system here? So antigen-presenting cell or tumor cell are presenting an antigen to the immune system here. So it might be APC with a virus or a microbe or a bacteria, of course, but it can be tumor cells with antigen from the tumor cells here. Activated T cells come now because the T cell is recognizing that something wrong is going on here. So what's going to happen here? So initially, the immune system is activated. We'll have production of interferon gamma and other cytokines. We'll have proliferation of T cells. And we have cytotoxicity against this APC or tumor cell presenting here, which is a main mechanism for fighting against infection, as an example here. So then the negative co-signal are going to come here. And the idea from the host immune system is avoid having a permanent activation of the immune system here. So as I told you before, PD-1 is one of the main co-signal which is going to make a break on the immune system here, PD-1, which is going to be important here because it will link with PD-L1, of course, if it's expressed, and it might be expressed in some tumor cells, in many tumor cells, in fact. And due to this interaction between PD-1 and PD-L1, negative co-signal, as I told you, this induced proliferation, cytokine activation, and cytotoxicity are going to go down, and then the immune system is going to become exhausted. That's what we call exhausted T cells. Immune response is going down, and of course, proliferation, cytotoxicity, cytokines like interferon gamma is dropping down. So why do we know that PD-L1 is important in tumors? And that's one of the first evidence that PD-L1 expression is important. That was reported in 2004 by the Mayo Clinic group, looking at PD-L1 expression in kidney cancer here. And you can see that PD-L1 is expressed in many RCC here. And interestingly, what has been shown by this group is that PD-L1 expression is a negative prognostic factor. The more you express PD-L1, the worse the outcome is. And that's probably due to the fact that the immune system is not able to fight against this bad tumor here, and explaining why RCC expressing PD-L1 have a poor outcome. And that's obviously not only in the localized tumor, but also in the metastatic tumor here. So that's what we have now. So we have an exhausted immune system, which is unable to fight against cancer. And that's why most patients develop cancer, and cancer grow and grow and grow. So the idea was to try to understand, because of this co-signal, can we block this co-signal, and can we restore the immune system here? And that's what we do when we block the interaction between PD-1 and PD-L1 with an antibody against either PD-1 or PD-L1. Just explain you why you can use both PD-1 or PD-L1 antibody in terms of this desactivation of the immune system. Both sides are working, and probably very similarly between PD-1 and PD-L1 antibody. The main difference is that we also have PD-L2, PD-L3, which are not blocked by PD-L1 antibody alone. But that's something that you have to understand. That's just blocking the interaction between PD-1 and PD-L1, between T cells and tumor cells here. And then we restore the activation. We restore the activity against the tumor cells, and of course we are able to restore cytotoxicity and immune sensitivity of the tumor cells. So as you know, I told you that this is not only the T cell and the PD-L1 expression. Mutational load is important here. And as you can see on this slide, which is a very important paper here, different tumors are different mutation rates here. And going from the left to the right, you can see on the right side some tumors which have a very high mutation load. And of course, some of these tumors are more sensitive to the immune system here. So that's a very important concept too, and maybe increasing the mutation rate might be a way also to increase the activity of the immune system here. And multiple factors are going to contribute to the generation of activated T cell immunity and tolerance. And you can see on this slide too, which is going to be some of the factors of the tolerance and some of the factors of the immunity. And that's going to be a kind of equilibrium and balance between these two factors to try to use the best we can the activated T cells and avoid tolerance. That's something that we have to avoid for fighting best to cancer here. So one of the questions now is, which are the immune biomarkers? And of course, that's one of the important parts of the work which is actually ongoing in many places, many institutions, to try to understand which are the predictive biomarkers here, which are the factors which are going to explain why immune system is working, why it's not working, how we could do better here. And of course, we have different parts of the expression here. PD-L1 expression in the immune system is important, both in tumor cells but also in T cell here. The blood vessels are important too, because if you don't have trafficking to the tumor cells, if T cells cannot go to the tumor cells because of vascularization, then the immune system is not going to work here. As well as we need antigen stimulation and presentation and activation. So we need T cells, lymph node probably, which are going to gather T cells, which are going to be able to activate the immune system. So these are different parts of the immune biomarkers which we are going to try to look at. We are going to try to understand better to predict which are going to be the factors to predict the good outcome for our patients here. So the final question for me is how can we modulate responses to immune system here? And that's something which is very important here. How can we do better than we do actually, as an example, with PD-1, PD-L1? And as you know, PD-1 and PD-L1 is actually involved in many tumor types with some good results in many solid tumors and even hematological malignancy, but we want to do better here. And we have different ways to increase the activity of this blockade here. So one way, of course, is to use another checkpoint inhibitor. And we have anti-CTLA-4 as one of the ways to use it, but we have a lot of other checkpoint inhibitor antibody which are going to come in the future, which are going to be in clinical trial in phase 1 here. We can also induce and increase trafficking of T cells by using some cytokines and trying to do better. We can use anti-VEGF therapy, which is very important both in tumors which express VEGF like kidney cancer, but also by increasing the infiltration of T cells by allowing better penetration of the T cells into the tumor here. And finally, by co-blocking PD-1 and PD-L1, there is some way, to try to combine PD-1 and PD-L1 blockade, which is also a way to do. Of course, we can also combine with chemotherapy. We can also combine with activated agents, targeted therapy, and that's a lot of approaches which are actually a way to try to modulate responses to checkpoint inhibitor here. So with that, I want to thank you. I hope that you will understand better the immune system now and will be able to follow the next session. Thank you very much.
Hello, my name is Ignacio Melero. I work at the University of Navarra Clinic. I'm responsible there for the immunotherapy program, both in research and in clinical practice. My job here today is to address biomarker discovery efforts in immunotherapy of cancer, a very challenging task. And also I will be covering immunotherapy of renal cell carcinoma. These are my disclosures. And actually, there are many things you can measure in a cancer patient trying to address whether the patient is going to benefit from treatment. In a way, the simplest dichotomy would be things that you can measure in the patient and things that you can measure in the tumor. And we have many tools. We can go to DNA and genomics. We can look at the messenger RNAs that are expressed. Actually, we can look at the microbiome, not forgetting that the immune system is highly regulated by the flora, the microbial flora. We can check parameters of immune function, both in peripheral tissues or in the tumor itself. We can use several tools of proteomics, and we can look at the epigenetics of the tumor, and all of those factors are going to really affect whether immunotherapy works or not. The problem is that this makes it very complex, and we certainly want to know what are the most predictive factors. Probably more than one factor affects a lot. This is multifactorial. So maybe we will do our best to assess the propensity to respond to immunotherapy by measuring several parameters. There is a review by Christian Blank in Science that I do recommend to address this multifactorial scenario. And in a way, they use a kind of a geometric figure whose area is going to reflect the likelihood of response. And several factors are plotted around this immunogram, as he referred to it. And one of the factors is tumor foreignness. We will go through it. The other one is whether the tumor is already infiltrated by immune system cells and what's the kind of immune cells that are there. Then the presence of the targets for the immune therapy treatments and the presence of the so-called checkpoints in the tumor. But there are other factors such as the microbiome, then there will be metabolic factors and also inflammatory mediators that we would like to measure. As I am mentioning, this is getting more and more complicated, but for clinical practice we need to make it simple. And simplification is often an oversimplification, but never forget that as it goes in the proverb, simplicity is the ultimate sophistication of complexity. And actually the role of many investigators now is to really identify what are the meaningful factors. So the first one I'm going to refer about is tumor foreignness, defined as the mechanism that provide the immune system with a target to identify the tumor and distinguish it from normal tissues. T cells recognize antigens in the form of peptides presented by self HLA antigen-presenting molecules. For HLA class I molecules, this presentation happens because of peptides coming from the transcriptome of the cell are loaded onto the binding cleft, peptide binding cleft of HLA class I molecules. If you look at this representation of the molecule from the top of the cell, you can see the peptide in red and the antigen-presenting molecule depicted in white and blue. And how do these peptides come onto these antigen-presenting molecules? Well, it's kind of a sophisticated pathway. The idea, and probably you know very well, is that the proteasome is constantly degrading misfolded proteins in the cytosol, and it breaks the proteins into peptides of different sizes, some of which are pumped into the endoplasmic reticulum by specific transporters. And once there, with a system of chaperones, they are loaded on this binding cleft of a nascent HLA class I molecule, which is mature in the Golgi and eventually comes to the cell surface, where it's presenting kind of the whole array of peptides fitting this class I antigen-presenting molecule. And for evolutionary reasons, this locus, which is expressed in a codominant fashion, is highly polymorphic. By that I mean that many different alleles can occupy each of the loci, that in the case of HLA class I molecules that present antigens to cytotoxic T lymphocytes are the locus HLA-A, HLA-B, and HLA-C. And as you can see, they can be occupied by quite a few different alleles. The reason for such a diversity is that they have evolved in humans and in apes in order to accommodate the most common pathogens, and there is a survival advantage if you can present dominant antigens from prevalent pathogens. But this complicates a lot the life of transplanters, but also of tumor immunology. An important thing to bear in mind is that if you look at the binding cleft of HLA class I molecules, there are several areas that are fitting amino acids of the peptides. Those are the so-called pockets that accommodate the lateral chains of some of the amino acids in the peptide. And because of that, there are patterns of peptides that can accommodate this binding cleft. These are usually, depending on the HLA molecule, 8 to 9 amino acid long peptides. And interestingly enough, when proteomic people started to elude these peptides and sequence them, they realized that there were shared patterns in such a way that suggested that you could make predictions of which peptides could be eventually accommodate a class I molecule. As you can see here in the picture, there are these anchoring amino acids represented in orange that are actually common in the many different peptides that are eluded from a given HLA class I molecule. And tumors, to be tumors, need to have mutations. and mutations would potentially give rise to antigens, to antigenic determinants presented by HLA class 1 or class 2 molecules, whether point mutations that are non-synonym in terms of protein sequence, deletions, insertions, would give rise to sequences that are not present in the normal transcriptome of the cell, and thereby can give rise to peptides, but not antigenic peptides, but not always, only when they do fit the endogenous repertoire of antigen-presenting molecules. Problems with RNA splicing or even epigenetic problems can eventually give rise to potential antigens. Ooh. So whenever a mutation happens, it can be antigenic for a number of reasons. One of them is that it will result in an amino acid that is not present in the native sequence, that it's phasing out, and it can be recognized by the antigen T-cell, the T-cell antigen receptor. Differently, it can also generate an amino acid or a set of amino acids that would fit better with the antigen-presenting molecule, thereby giving rise to a neoepitope or a neoantigen. And then if the mutation is more complex, several of these things can happen together. But those are the basis for antigenicity in tumors. The interesting thing is by the group of Tim Chang in the Memorial Sloan Kettering Cancer Institute. It became clear that the more mutations a tumor had, in this case in metastatic melanoma, the more likelihood to clinically benefit from a checkpoint inhibitor, in this case blocking CTLA-4 with the antibody ipilimumab. But not only mutation burden. If now they apply algorithms to predict the likelihood of these mutations to fit with the HLA alleles present in the genome of the patient, then the correlation with survival became even more clear. So it's possible that the main factor linking antigenicity with tumor burden and prognosis or predictive value for immunotherapy is related to the number of epitopes that are actually, the neoepitopes that are actually presented by tumor cells. This seems also to be true for non-small cell lung cancer. The pioneer group work by Dr. Ritzby in this regard showed that there was a correlation of the clinical benefit from pembrolizumab in these patients depending on the mutation burden. And again, when neoepitopes were predicted in silico, there was a good correlation between overall progression-free survival in this case and of the patients following pembrolizumab treatment. Interestingly enough, using the tricks that immunologists used to actually be able to see that the patient has T cells responding to these antigens, it was possible to see an increase in the immune response as a result of PD-1 blockade, and it was possible to see that some of the epitopes were neoepitopes were actually recognized in the patient, because if you got those T cells and exposed them to synthetic versions of the antigen, they started to produce gamma interferon that can be measured by intracellular staining, and that's what these dot plots here show, is that some of these antigens were actually recognized by larger numbers of T cells in the patients. Another important point coming from the Cree Institute here in London, in this paper by McGranahan et al. Published in Science, is that they used a trick to actually be able to tell whether the mutation was present in all the tumor cells or it was heterogeneous. is what in tumor genetics they call an stem mutation, which is there probably all the way from carcinogenesis, or what is a branch mutation that is not shared by all the tumor cells. And as you would probably have predicted, those patients in which the neoantigens are in the stem, are in all the tumor cells, are more susceptible to immunotherapy than in those cases where there is multiple clones of tumor cells expressing different mutations. And this is probably due to the fact that all the tumor cells are expressing on the antigens, which are ultimately the target of the immune response that we exploit in cancer immunotherapy. Indeed, using these algorithms to predict heterogeneity in the expression of antigens, they could see a nice correlations to tumors. These are not easy biomarkers at all because they presume that you have to do whole exome sequencing of the tumor, normal tissue, typically peripheral blood, compare them, and then do a lot of bioinformatics in order to predict the epitopes. But if it were a good biomarker, eventually costs of sequencing and of bioinformatics can be dropped, and this could be done in the future. Another very important point, and this was realized by Tom Gajewski in the University of Chicago, and also by Franco Marincola at the NCI, as that the tumors that tend to respond to any kind of immunotherapy, whether vaccination, immunomodulation, whatever, tend to be those that have already an infiltration of T cells in the tumor, in such a way that immune infiltration in itself is very often a prognostic marker, a favorable prognostic marker in tumors. But, and this is an interesting trick, what probably immunotherapy does is to unleash an immune response that is already ongoing. And unless you have an immune response already ongoing, then immunotherapy tends to be not successful. In this way, we can classify tumors as hot and as cold. Those tumors that we call cold tumors are tumors that do not have T cells inside. Then we have tumors with evidence for pre-existing immunity, particularly those with a lot of CD8 cells or T cells producing interferon gamma. Those are what we call hot tumors. And then obviously you have a spectrum where some of them are colder than others. And there is something also that we see a lot is what we call the excluded infiltrate, and those are tumors with an invasive margin with T cells, while the malignant tissue itself, where the tumor cells nest, is devoid of T cells. And what defines a hot and a cold tumor, it's very complex. Actually, a lot of the research right now in cancer immunology is trying to address precisely this point, because there are factors that are related to the genetics of the tumor, to the epigenetic of the tumors, with the kind of inflammatory response that it's there. And actually, it would be kind of very difficult to generate a very good algorithm to predict these. Things are getting very complicated, as you can see here, and if you consider all the functions that you need for a successful immune response, virtually, you could be measuring all of them and trying to figure out whether or not this is giving rise to an effect on immunotherapy. But at the end of the day, what has already been successful is that we know that checking the transcriptomic signature of genes that respond to interferon gamma correlates with propensity of response. There are several signatures that have been validated and patented, and they are routinely used. This is simple because it's a simple macroarray that can be done. Then the evidence for activated CD8 T cells, if they have the molecular means that they use to kill, such as perforin or granzyme B, and then the type of macrophages that are inflammatory and are producing a kind of the right type of mediators, such as TNF-alpha. Those are what we call M1 macrophages, and they are typically elicited also by interferon gamma, and they are nice guys for immunotherapy, as opposed to what we call M2 macrophages, which are producing angiogenic factors and growth factors, and the kind of inflammation driven by IL-6, for example, that it's not a myeloid inflammation, that it's not good for immunotherapy. Obviously, in a population of cancer patients, we get these parameters in different fashion. It could have kind of a bell-shaped curve, and some of them are more propense to immunity, some of them more to tolerance. Genetics, age, the microbiome, the presence of previous viral infections, the exposure to carcinogens, the exposure to all that defines this propensity, and then it's very multifactorial. But the methods that we typically use to analyze all these are conventional immunohistochemistry. We are learning now that it's much better to have more than one parameter information in the tissue section. And for that reason, multiplex immunofluorescence techniques are becoming widely used, and actually they provide a lot of information. And since fluorescence is better than chemistry with regard to quantitative measurements, immunofluorescence can provide semi-quantitative assessment of the level of expression of the different proteins. Using RNA-seq or different forms of RNA expression techniques of multiple genes in a single run are also very important. And as I mentioned, and mainly because of analyzing the foreignness of the tumor, the number of mutations and their likelihood to give rise to neoepitopes, we use whole exome sequencing. And we are trying to learn also whether there are genetic or epigenetic features of the tumors that are mediating this immune exclusion, something that has a lot to do with the vascularization of the tumor and the type of microvasculature that it's in the tumor. A very interesting suggestion came again also from the group of Tongajewski, and that comes from the fact that cytotoxic T lymphocytes, CD8 T cells, typically to get them turned on, you need to present antigen with a very interesting subset of dendritic cells, of antigen presenting cells, or professional antigen presenting cells. This small subset is characterized by expressing the transcription factors BAT3 and also IRF8 and in a way, what we have learned more recently is that at least in melanoma, but probably also in many cases in the TCGA, it comes a very interesting feature is that hot tumors tend to be richer in this particular subset of CD103 dendritic cells than those who are not. And even then, you could see hot tumors in red, cold tumors in blue here, and you set your cutoff in the middle, and it's interesting because this correlates better with the presence of these professional antigen presenting cells for CD8 cells than actually the mutation burden that is represented here in the upper graph. So it's probably the case that it's not only gene mutations what make tumors recognizable by the immune system, but also, and importantly, the presence in the tumor of these antigen-presenting cells. And actually, those tumors which are immune deserts that are not populated by T cells are probably primarily devoid of these antigen-presenting cells in the infiltrate. We don't know what's first, the chicken or the egg, but this is a field that is going to be very active in the next 5 years. An important practical knowledge is that B7H1 is another name for PD-L1, and the presence of TILs can be combined together. And by the different combinations, you can get 4 phenotypes, 1, 2, 3, and 4 here. Some of them which are deserts, do not have PD-L1 or tumor-infiltrating lymphocytes. Some that have PD-L1 but not TILs. Those that have both of them expressed, and some of those which only have TILs but not PD-L1. And this is probably a very important thing because we can personalize treatment for each of these subsets of patients. Those who have an immune response going on and PD-L1 is there probably blocking the response are going to be those patients who are more likely to respond, as opposed to the immune deserts where probably we will have to do something else in order to get an immune response against the tumor, something like vaccination or a co-treatment that would enhance this infiltration by immune system cells. So I guess that now you are going to watch this video about PD-1 and PD-L1 blockade from a molecular perspective.
In recent years, immune checkpoint blockade has taken centre stage as a significant breakthrough in medical cancer therapy. Activation of T cells involves several paired molecular interactions, such as T cell receptors or CD4 with the major histocompatibility complex, B71 with CD28 and several others. Programmed cell death 1, or PD-1, is a member of the CD28 superfamily and is expressed on T lymphocytes. PD-L1 and PD-L2 are its ligands on cancer cells. Mechanistically, T cells use PD-1 to probe cancer cells for PD-L1 and PD-L2. When they find it, they leave tumour cells alone to survive. When PD-1 binds to its ligand, it induces immune suppression in the tumour microenvironment, which subsequently leads to resistance of cancer cells to the immune system. However, when a molecule blocks this identification, T cells are no longer misled by the presence of PD-L1 and PD-L2, and subsequently rally an immune system attack on the cancer cell. Antibody-based PD-1 or PD-L1 blockade has achieved strong success in clinical trials. These agents take advantage of specific binding to antigens and thus have been widely used for immune checkpoint blockade. Multiple PD-1 or PD-L1 antibodies have been approved for clinical use or have entered into clinical trials, including nivolumab, pembrolizumab, atezolizumab, durvalumab, and avelumab. The first clinical trial of a PD-1 blocking agent began nearly a decade ago in patients with blood cancer. Today, more than a dozen trials have been completed, with more than 50 underway, involving thousands of patients with several different types of cancer. These ongoing trials are encouraging, particularly since they indicate that a substantial number of participants have benefited, resulting in a need for further trials. Additionally, they have suggested that these agents may be effective against multiple types of cancer.
Having seen that video, it becomes important to know whether the tumor is expressing PD-L1, whether there is PD-L1 expression in what we call the tumor microenvironment. And there are several agents blocking either PD-1, nivolumab, pembrolizumab, or PD-L1 such as atezolizumab, durvalumab, or avelumab. And it's not easy at all to detect PD-L1 expression on formalin-fixed and paraffin-embedded tissue. There were many attempts, many cases of failure to generate good antibodies staining this moiety in the tumor tissue. And there are several issues about epitope retrieval, then the folding of the protein, and other factors that makes this very, very difficult. In such a way that the companies developing these compounds have invested heavily in developing or buying reagents that would reliably quantify the number of cells that are expressing PD-L1 above a certain cutoff. these companies have developed their antibodies and automatized the detection and and how you quantitate the number of cells. And you have here several antibodies, 28A8, 22C3, SP142, and SP263. And importantly, since these compounds are being developed in the same period of time, it has become very important to actually be able to compare what's the result with the different antibodies. It is also very difficult to have the pathology departments running all the tests and all the platforms for detection. In most cases, the technique is only taking care of expression with a surface pattern on tumor cells, and then you quantify a brown cell and cells that are not stained, and you end up with a percentage of expression. There's one exception, is that atezolizumab being developed by Genentech, they were more interested in checking not only the expression on tumor cells, but also on immune cells infiltrating in the tumor, since they found a correlation with the likelihood of response, of objective response to atezolizumab, at least in some types of tumors. In this case, they generated a score depending on expression on tumor and expression on tumor cells. And then they studied different cutoffs in the different studies, using it as a predictive biomarker. As you can see in several attempts to see the consistency of staining with the different compounds, you can see that most of them have a nice correlation. One of the problems is in the steepest part of the curve of correlation, where very little differences can mean a lot, and probably is where the cutoffs are. And in the case of SP142, it disclosed less positive cells, but remember that this antibody was generated probably mainly to detect expression on immune cells. This is how the thing looks like. So as you can see, is not that easy. These are typical examples. So when it is 100% of the tumor cells, it's easy, but then it becomes a little bit less cumbersome, because staining is typically patchy and tends to concur with the areas where T cells are. And one of the reasons for that is that the main inducer of PD-L1 expression is interferon gamma, which is a cytokine produced by tumor-infiltrating lymphocytes if everything is going okay. And then the predictive value as a biomarker after this complex detection in the pathology department is good, but it's not great. By that I mean that you can enrich for patients who would eventually respond to PD-1 or PD-L1 blockade. But as you can see, depending on the disease, this level of enrichment is twofold in the best of the cases. And that's probably not enough to negate treatment to people who have no other treatment with known efficacy. So we really need to get better and beyond PD-L1 staining in the tumors. And again, we go to this nomogram, trying to see what else we can try to plot together in order to have a better picture, knowing that the complexity is great. And there is something which is emerging as very, very interesting, and that's the presence of bugs in the gut, potentially also in the skin. As you know, the microbiome is a huge collection of bacteria that nest in the gut, especially in the colon. And with many species, and that we know for a long time that they regulate immunity quite a lot. In fact, it's species specific as well as a healthy presence of bacteria, giving rise to important components in the pathogenesis of autoimmunity, for example. In fact, the gut microbiome could be not only a prognostic or predictive factor for immunotherapy, but also potentially a predictive factor for immune-related toxicity in cancer patients, because there are many mechanisms that are regulated by the presence of some components of the microbiome that go to the level and degree of infiltrate in the tumor and are governed by controlling the function of dendritic cells at this level, but also systemically. If you are curious about this, I do recommend some reviews, including one by Lawrence Bohel and Tom Gajewski, who were the pioneers showing in mice that the microbiota actually regulated the immune response against cancer. And not only the basal immune response, but also, and importantly, the response to PD-1, PD-L1 blockade or to CTLA-4 blockade. Another important issue is resistance. Immunotherapy is not going to be an exception to other cancer therapies. Tumor cells can develop resistance or be resistant to begin with. Resistance in tumor cells can be present in all the tumor cells in the tumor mass. Maybe some clones will adapt. Maybe at the end of the day, you could have a very, very nice response, but persisting cells would adapt. And the molecular basis for these are only being started to be studied. This is mainly because immunotherapy was not very effective for a number of years. And it is since the first decade of the 21st century that we are achieving some degree of efficacy, mainly in melanoma, that now extending to other diseases. So in the absence of efficacy, we did not worry that much about acquired resistance. And resistance, and this field has been pioneered by the group of Tony Rivas in UCLA, can be primary depending on genetic and epigenetic features of the tumor. For example, several oncogenes make the tumor less likely to be inflamed, to be hot. But tumors can escape also by downregulating the antigen presentation machinery that I introduced to you at the beginning of the first part of the lecture. For example, downregulating the molecules needed for antigen processing and loading, and importantly, the antigen presenting molecules itself. In fact, HLA class 1 molecules are stabilized. They need to be stabilized by a protein called beta-2 microglobulin, which assembles and stabilizes the complex. And in the absence of beta-2 microglobulin, there's virtually no HLA class 1 molecules on the cell surface. And in that setting, a complete loss of beta-2 microglobulin would give rise to a tumor that's invisible to CD8 T cells and thereby would provide an escape route. HLA class 1 molecules are typically expressed at a very low level. And only when there is type 1 interferon or gamma interferon around, where there is signals that are denoting a viral infection, is when these gets expressed at a logarithmic scale, several logs of increased expression. So precisely this pathway of signaling from the interferon gamma and the type 1 interferon receptors can be down modulated by mutations or some kind of epigenetic adaptation, and if that happens, there will be a much decreased level of antigen presentation in the tumor. And that's precisely which seems to be a major route of escape precisely in melanoma. The type 1, and in this case the interferon gamma receptor, relates on two tyrosine kinases that get associated to the cytoplasmic tail, JAK1 and JAK2, that activate the STAT1 and STAT2 transcription factor that relocates to the nucleus after phosphorylation and regulates many genes, including, importantly, HLA, MHC, class 1 expression, and PD-L1. If the tumor happens to mutate or get rid of the expression of JAK1 and JAK2 or one of them, there will be this pathway will stop. And that's precisely what Tony Rivas reported in several patients with acquired resistance to PD-1 blockade in melanoma. And I'm going to tell you a little bit a particular observation that we are proud of in our institution, which is using interleukin-8 as a biomarker. We recently published several papers in clinical cancer research and Annals of Oncology about it. The interesting thing is that IL-8 is produced mainly by malignant cells, and in the cancer patient peripheral blood, often you can detect high levels of IL-8, which is a chemokine that attracts myeloid cells, particularly neutrophils, to the tissue where it is being produced. Interestingly enough, if you xenograft mice with tumors, with human tumors, immunodeficient mice, of course, what happens is that the human tumor produces IL-8 that you can detect in the peripheral blood. But interestingly enough, this gene is not conserved in rodents in such a way that all the IL-8 you measure comes from the tumor. And as long as the tumor grows, you start detecting more and more IL-8, and if you perform a surgery and remove the tumor, the IL-8 levels drop. So you can use this as a surrogate marker for tumor volume. And this actually works as that. So the patients with heavy burden have more IL-8 than those who do not have a heavy burden. But more importantly, what we found is that patients who were treated with PD-1 blocking agents, if they eventually responded, we see a decrease in IL levels as opposed to the peripheral blood samples of those patients who do not benefit from treatment and the disease progressed, and this was true in melanoma, in non-small cell lung cancer, also in hepatocellular carcinoma. Interestingly enough, if the parameter that you measure is not the absolute value of the concentration, but instead of that, the change, whether it goes up or goes down, then the predictive value becomes quite reliable. And although these series of patients are not that large, this is being prospectively validated in large serum collections. And of course, this is not a predictive biomarker because you assess it 4 weeks after, 3 weeks after starting treatment. But don't forget that we very badly need this kind of biomarkers early on treatment so we can save cost, we can save side effects, and also we can save time in order to get another line of treatment to the patient early on if we already can define the patient early on and probably go to another clinical trial or whatever other therapeutic option. Another interesting thing is that in immunotherapy, as you have learned in previous talks, it's treatment modality that can result in what we call pseudoprogression due to tumor inflammation that by imaging techniques is wrongly interpreted as a tumor progression, while what is really happening is an immune response. This is not that frequent, but often is the case that you doubt on the first attack, on the first CT scan, that the patient is not responding. So in this case, what we found is that IL-8 seems to be good at telling apart those cases of true progression versus pseudo-progression, as happened, for example, in this case of a patient with bladder cancer treated with atezolizumab. Again, this correlates with survival following treatment. So to sum up, antigenicity, immunogenicity by proper antigen-presenting cells, presence of mechanism of immune escape in the tumor, presence of T-cell infiltrates pre-treatment, and immune signatures, particularly those related to interferon gamma expression at the tumor tissue, are supposed to be the main ones we are going to rely on from a practical point of view. Bear in mind that PD-L1 determination in tumor tissue as a single parameter is not good enough, although it's helpful, and probably will let us stratify patients very well in clinical trials. And certainly we are going to go to these multi-parameter type of scores, including the tumor PD-L1 status, but not only. And probably we will have to go to more quantitative measures rather than just the expression, the number or the proportion of tumor cells expressing PD-L1 on the surface. So now we are going to switch gears and we are going to speak about immunotherapy of renal cell carcinoma. As you know, this is a terrible malignancy. It's very often diagnosed in an advanced stage, and it's a tumor which is immensely vascularized. It's a tumor with the hypoxia signal on most of the time, and it has many vessels in its microenvironment. It is a very interesting tumor because in the previous part of my talk, I mentioned to you that there was a tendency in the sense that those tumors which have an important number of T lymphocytes, pre-treatment on them, tend to be those which respond to immunotherapy and those with a better prognosis. Actually, in some tumors like colon cancer or ovarian cancer, this is an incredibly good prognostic factor. But in renal cell carcinoma, for whatever reason that we don't know, things are actually the other way around. Those tumors with a kind of denser T-cell infiltrate of T-cells producing interferon gamma fare worse in terms of prognosis. We don't know that. We don't know the reason for that, but it's something that will make this tumor kind of peculiar and interesting with regard to immunotherapy. However, when the first antibody against PD-1 got into development in a landmark phase 1 clinical trial, nivolumab, disclosed its very first data, it became very clear that this was a tumor that was amenable to immunotherapy, with an important number of patients developing responses to treatment in the different dose level cohorts of these phase 1b clinical trials. And importantly, responses, when they did occur, tended to be durable. In a way, this was one of the tumors that was selected together with melanoma, metastatic melanoma, and with non-small cell lung cancer, because there was previous evidence of a response to immunotherapy. Actually, treatment with high-dose IL-2 and with interferon had shown signs of clinical activity and even gave rise to approvals in this disease setting. Prompted by these results, there was a phase 2 trial, was a randomized phase 2 trial testing 3 different dose levels in a randomized fashion, and patients were treated until progression of or intolerable toxicity. I will tell you later why that's important. And then there were very strong signs of long survival, medians of survival beyond 2 years. And in the 3 dose levels, but I must tell you that in these 3 dose levels, probably we are achieving full receptor occupancy, at least in peripheral blood. So these data were really, really encouraging in second-line metastatic renal cell carcinoma. Hence, a phase 3 pivotal trial was designed by the same company and was launched testing nivolumab in second-line treatment of metastatic renal cell carcinoma in comparison to the most often used second-line treatment, which is the mTOR inhibitor everolimus. And the primary endpoint was overall survival. Interestingly enough, there was a good number of patients, going up to 25% of these patients, who experienced responses by RECIST 1.1 criteria, which was really encouraging. Actually, this clinical trial was halted by the disease monitoring committee because of superiority of treatment. And actually the responses, when they occur, tended to be durable, with many of them ongoing at the time of analysis, and clearly superior to everolimus. The curves, the Kaplan-Meier curves depicting survival were very interesting for second line, although I must say that we were surprised by not seeing a plateau of patients, of the curve eventually talking about a very long-term patients benefiting from treatment. Nonetheless, this is a clear positive phase 3 clinical trial. Although, importantly, it would have been missed if progression-free survival would have been used as the primary endpoint, since no difference was seen there. So a lot of patients were developing stable disease, and that was probably part of the reason of the extended survival. From the point of view of the waterfall plots, you can see that this is really interesting for patients with metastatic renal cell carcinoma with a slow progression or tumor shrinkages as the best response that were not that would be underestimated by the overall response rate. In fact, another very important observation was made is that some of the patients were treated beyond progression and some of them were not. And when you dichotomize for these patients, it looks like treatment with PD-1 blockade beyond the point of time when radiology progression was called benefited the patients, as you can see in this comparison. From the point of view of safety, there is nothing really to add to what you have already seen in other presentations of immunotherapy, and in fact, probably there was a better tolerability in many of these patients, perhaps with pneumonitis being the side effect that we are more fearsome in the cases that were treated in this pivotal trial with more than 400 patients treated in the nivolumab arm. So following this pivotal trial, this is the scenario of how we guide treatment of metastatic or locally advanced renal cell carcinoma that is not surgically amenable. First line of treatment has several options, including, importantly, clinical trials, and more importantly so, clinical trials of immunotherapy. But now we have sufficient degree of evidence to indicate nivolumab as a second-line treatment. We don't know yet if it's better to use it in any other disease setting, but certainly it's useful in second-line treatment of renal cell carcinoma. Atezolizumab, as a PD-L1 blocker, has already been tested in phase 1-2 clinical trials for renal cell carcinoma. As you can see here, there is activity going to a nice response rate that, interestingly enough, seems to not to correlate with the real degree of PD-L1 expression in the tumor. It's a very, very, very small correlation. Importantly, patients treated tend to survive very much. I mean, these curves for second-line renal cell carcinoma are truly interesting, although we don't have comparative clinical trials yet. Immunotherapy with anti-CTLA-4 antibodies, in this case with ipilimumab, has also signs of clinical activity, and there are cases who responded to this CTLA-4 blockade, although they were a minority. However, this was the clue to start combining this drug in several settings. In fact, ipilimumab is being combined with nivolumab in a multi-arm clinical trial, which is extremely interesting. And even in this kind of phase 1, phase 2 setting, it's clear that seeing responses that go up to 40% of the patients is nothing that has been seen before in the treatment of second-line metastatic renal cell carcinoma. Another thing to consider is that vascular endothelial growth factor, that many of us have studied very well as the main driver of tumor angiogenesis and vasculogenesis, is probably also a very important immune suppressor. There is plenty of evidence that abundant VEGF presence in the tumor microenvironment actually decreases the level of immune cell infiltrates in the tumor and their function. In early observation, it was found that atezolizumab plus the VEGF blocking antibody bevacizumab has activity that looks promising and potentially giving rise to synergy in metastatic renal cell carcinoma. And in this trial, it was very interesting to see that when vascular endothelial growth factor was blocked, there was a more prominent infiltration of T cells in the tumor with higher levels of expression of MHC class 1 molecule and PD-L1, probably as a result of local release of interferon gamma. And thereby, it looks like there is synergistic activity when using inhibitors of the VEGF receptors, type 1 and 2, together with the different agents blocking PD-1, PD-L1, such as nivolumab, avelumab, pembrolizumab in this case, using different compounds known to inhibit the tyrosine kinase activity of these VEGF receptors. Therefore, this is really an interesting option to be further explored in this disease. And in fact, there are many ongoing clinical trials testing immunotherapy in first-line renal cell carcinoma. Some of them are already fully accrued in such a way that we are expecting results soon, and it could be the case that some of these combinations, remember that anti-angiogenic compounds are the currently most used treatments in first-line renal cell carcinoma, but all these trials are really addressing the point of seeing whether in first-line addition of PD-1, PD-L1 blocking agents would would give rise in a comparative fashion to a better outcome. Remember, we are at the beginning. This is really the beginning of clinical exploitation of immunotherapy. We are very much in need of good biomarkers, and we certainly expect progress soon in renal cell carcinoma immunotherapy. Although, as you've seen, there is a strong case for using this treatment modality in the second-line setting. I hope that during the talk I stress enough that we are at the beginning. This is not the end, probably, and this is not even the beginning of the end. And to guide us, we are very much in need of biomarkers, particularly predictive biomarkers. And then the second message to take home is that we expect progress in renal cell carcinoma immunotherapy in the very near future, also in the direction of biomarkers as well. But as you've seen, there is a very strong case for using immunotherapy with nivolumab in the second-line setting. So thank you very much for your kind attention. And the take-home messages are that we are at the beginning of something rather than at the end of something, or even at the beginning of the end of something. And immunotherapy is here to stay, and it's in very bad need of biomarkers, particularly predictive biomarkers, as we've seen during this talk. And then an important message is that expect progress soon in renal cell carcinoma immunotherapy. It is really something that is going to come in the near future. But remember that it's already reality in the case of nivolumab for second-line treatment of renal cell carcinoma in a metastatic stage where it has a very profound effect. So thank you again for your attention, and I will leave you now with the next program, which is efficacy and safety of immune checkpoint inhibitors in non-small cell lung cancer. This has been a revolution of the PD-1, PD-L1 pathway as well. This lecture will be given by Dr. Peter Schmid. Thank you very much.
My name is Peter Schmidt. I'm a medical oncologist and work at Barts Cancer Institute in London. I lead the immune therapy program there, looking after patients with lung and breast cancer. It's my pleasure to talk today to you about the efficacy and safety of immune checkpoint inhibitors in non-small cell lung cancer, with a key focus on PD-L1 and PD-1. As we know, for immune therapy, there are a few things that need to come together. On the one hand, we need to have neoantigens which are linked with mutations. On the other hand, we have to have tumor-infiltrating lymphocytes that can be activated and then tackle the cancer. If you look at lung cancer, as you can see on this slide, lung cancer, squamous cell lung cancer as well as non-squamous cell lung cancers are characterized by a higher rate of somatic mutations compared to many other tumors. So from that point of view, non-small cell lung cancer has always been a good candidate for cancer immune therapy. If you look a bit more into the biology of non-small cell lung cancer, you can see there are very distinct groups depending on the immune infiltrate. There's a group with high PD-L1 expression on the tumor cells, and there's a separate group with high PD-L1 expression on immune cells. These groups have very different biology in the way the immune system is activated and behave very differently clinically. On the other hand, when it comes to cancer immune therapy and treatment with immune checkpoint inhibitor, and in this case with atezolizumab, we can see that both groups, tumors that have a high tumor cell expression of PD-L1, as well as tumors that have immune cell infiltration with PD-L1 positive cells, respond equally well. So the subtype of immune expression seems to have less relevance for the response to immune therapy, but it's important to recognize that PD-L1 expression is not just on tumor cells, but also in immune cells in non-small cell lung cancer. If you look at the very first data we received with immune checkpoint inhibitors in non-small cell lung cancer, we learned very early that patients have substantial responses to single-agent therapy, whether it was with atezolizumab, pembrolizumab, or nivolumab. I would like to share one of the cases of one of the first trials we did with cancer immune therapy in non-small cell lung cancer. This is a case of a 66-year-old lady who is a former smoker with a history of 3 packs per day for 35 years. She was diagnosed in 2011 with metastatic lung cancer and metastatic involvement in lung, bone, soft tissues, and brain. Her molecular analysis showed that the tumor was wild type for EGFR and ALK. She had her solitary brain metastasis treated with stereotactic radiotherapy, then received as first-line treatment standard chemotherapy with pemetrexed and carboplatin, followed by consolidating radiotherapy. She progressed just at the end of 2012 and had second-line chemotherapy before she progressed again and was enrolled in a single-agent study with the immune checkpoint inhibitor atezolizumab. As you can see, at baseline, her disease was widely spread, as you can see on the PET-CT scan on the left side and the CT scan on the right side. Tumor was strongly positive for PD-L1, as you can see on the slides up here, with strong expression of tumor cells for PD-L1 in this lady. When we treated the patient, she had a very quick symptomatic improvement, and her scan after 3 cycles, as shown on the right side, shows a dramatic response of her tumor manifestations on PET-CT, and this was confirmed on her CT scan. So it's a classic example of a patient who responds very, very quickly, has a dramatic response to immune checkpoint inhibitor. And these early studies were the basis for a whole series of randomized phase 3 studies that explored the efficacy of different single-agent PD-1 or PD-L1 targeting treatments compared to docetaxel as second or third line chemotherapy in patients with advanced non-small cell lung cancer. Patients in these trials had to have progressive disease after at least one line of prior platinum-based therapy, and patients with EGFR mutant tumors or ALK rearrangement had to have received targeted therapy. I'm presenting all these studies together. These are studies involving nivolumab in CheckMate 17, which is a study focusing on squamous cell cancers, nivolumab in CheckMate 57, another randomized phase 3 study focusing on non-squamous histology, the OAK study with atezolizumab that combined squamous and non-squamous tumors, and the Keynote 10 study, which had only patients with PD-L1 positive tumors and they were treated with pembrolizumab. If you look at the first endpoint for this trial, overall survival, you can see that in all these studies, overall survival was positive. There are differences in how early those curves separated, but essentially in all studies there was a hazard ratio between 0.59 and 0.73. All studies met the endpoint that single-agent immune checkpoint inhibitor therapy, whether it was targeting PD-1 or PD-L1, was significantly superior to second-line chemotherapy with docetaxel. There was a long discussion initially whether patients with squamous cell cancers would benefit differently and possibly better than patients with non-squamous histology. But again, as you can see here from the OAK study, there was no difference in the hazard ratio for patients with non-squamous cancers or patients with squamous cancers, hazard ratio for survival being 0.73 in both subgroups. Similar results were observed within the Keynote 10 trial. So at this point in time, we could not see a clear difference between squamous cell cancers and non-squamous cell cancers, and most studies going forward are allowing patients with both histologies in the same trial. One interesting question is there a difference in benefit in terms of overall survival depending on the PD-L1 expression? As you can see, these trials had PD-L1 negative patients in there, patients with PD-L1 positive tumors, and the data here are shown by different cutoffs. PD-L1 positivity defined as 1% or higher, 5% or higher, 10% or higher, or a subgroup of patients with 50% or higher. I think what we can see from this slide is in all subsets, PD-L1 high, PD-L1 positive but not high, or PD-L1 negative, in all subsets, patients did derive a substantial benefit in terms of overall survival if they were treated with an immune checkpoint inhibitor in second or third line treatment as compared to docetaxel. We can discuss whether there may be a slightly higher numerical benefit for patients with higher PD-L1 expression, but the variation between trials is significant there, and I don't think we can make this statement very clearly at this point in time. What about progression-free survival? And progression-free survival is slightly different if you look at the different trials. As you can see, in some trials there's early separation, and if there's early separation of the curves, that led to a clear benefit in progression-free survival, as you can see in the CheckMate 17 trial for squamous cell cancers. In other studies, there was initial crossover, which means initially patients with chemotherapy had a slight benefit, but later on there was a crossover. And as you can see in all trials, there's a tail of the curve. If you wait long enough, patients with immune checkpoint inhibitors will have a benefit in terms of progression-free survival, but it depends on how early those curves separate, whether those results were positive comparing the overall curves between docetaxel and immune checkpoint inhibitor therapy. Now, what is interesting is looking at PD-L1 expression and PFS. And as you can see on the left of the slide, these are patients with PD-L1 negative tumors. And in these tumors, the data are obviously not consistent between the trials. But I think if we were taking an average of those three trials, there's no clear benefit in terms of progression-free survival for PD-L1 negative tumors. Whereas if you focus on PD-L1 positive tumors, and especially on tumors with higher expression of PD-L1, there seems to be also a benefit in progression-free survival. Some of this in progression-free survival compared to docetaxel is dependent on how well patients respond. And if you look at the response rate here of nivolumab versus docetaxel, we can see overall the response rate of immune checkpoint inhibitors is also higher compared to chemotherapy. And more importantly, the duration of response with 17 months compared to 5.6 months, again, is significantly better. But the important question here is how does response and PD-L1 expression, how are those two things connected? And if you look at the data from across those trials from Keynote 10, Checkmate 17, Checkmate 57 and OAK, you can see there are differences. There are differences in response rate. In Keynote 10, in the group with 50% or higher PD-L1 expression, the response rate of a single-agent immune checkpoint inhibitor therapy is very high, with 29% compared to 8% with docetaxel. Whereas if you look at all comers in OAK, which includes PD-L1 positive and negative patients, there's barely a difference between chemotherapy and atezolizumab in this study. And if you look on the right side of the slide, depending on the amount of PD-L1 expression either on tumor cells or immune cells, with atezolizumab from the OAK study, the higher the PD-L1 expression is, the bigger the difference in response rate observed between chemotherapy and immune checkpoint inhibitor. In other words, if there's high PD-L1 expression, tumors tend to respond better to immune therapy, whereas if there's low or absent PD-L1 expression, then the response is still there, but the response is no longer significantly better compared to docetaxel. And taking this into consideration, this will explain the differences in PFS analysis between the different studies. Because some of the studies like OAK, where there was little difference in response rate, is also a study that did not show a significant benefit in progression-free survival. Whereas if you select other studies that have more patients with higher, for example, PD-L1 expression, are therefore more likely to respond significantly in terms of an objective response to immune checkpoint inhibitor therapy, the studies are more likely to show a difference in PFS. The interesting observation is when it comes to overall survival, response does not seem to be that relevant. In other words, patients can have an overall survival benefit even if there's no clear difference in response rate. But when we need to achieve an objective response, then patients who have higher expressing PD-L1 tumors are more likely to have a substantial response of immune checkpoint inhibitors. Now, with the substantial benefit of single-agent immune checkpoint inhibitors in second and third line therapy for non-small cell lung cancer, the key question is, how well do these agents work in first-line therapy? If we look at first-line therapy, it becomes also more relevant to differentiate the different subtypes of non-small cell lung cancer. And if you look at the expression of or the infiltration of tumor-infiltrating lymphocytes in non-small cell lung cancer, we see there's a group that's highly infiltrated with immune cells that is what we call a hot or an inflamed tumor type. These tumors are often characterized by high expression of PD-L1 checkpoints, by high infiltration of CD8 positive cells, by an activation of those cells as measured by an interferon gamma signature, by higher mutational load and by higher TILs. On the other hand, we have other tumors where there's still immune cells around, but these immune cells seem to be around the tumors and can't infiltrate into the tumor itself. And this is a group called excluded infiltrate. There's a third group of tumors that are relatively cold. They have low amount of T cells and often low MHC class 1 and are often differently in terms of their proliferation rate. As you can imagine, the immune therapy strategies for these tumors should probably be different. Tumors that are inflamed or hot tumors may be excellent candidates for single-agent immune checkpoint inhibitors, and we have data, and we'll share data with you in a minute, for patients that fit this category with non-small cell lung cancer diagnosis. On the other hand, patients with excluded infiltrate, we need to work on combinations to bring those tumor-infiltrating lymphocytes back into the tumor, and these combinations can, for example, with CTLA-4 or other partners, or combinations with vascular targeting agents, or, for example, MEK inhibitors. And possibly with this, we can increase the infiltrate and therefore then in a second step activate those immune cells and have an immune response. More challenging is how to treat patients with cold tumors, and we need to work on priming and activating those, the immune system there and leading to better infiltration, possibly also modulating the neoantigen expression, but this is not achievable in general with single-agent immune checkpoint inhibitor therapy, and again, these tumors should be considered for combination therapy. In terms of combination therapy, what is important is to understand the whole immunity cycle that happens, or the interaction of cancer and the immune system. And the immune system the effect of the immune system starts with antigens being released of cancer, and of course, if you, for example, have more cell kill through radiotherapy or chemotherapy, then it can lead to increased antigen release. These antigens need to be taken up in antigen-presenting cells, be presented to the immune system. It leads to priming and activation of immune cells, which are then transported to the tumours, infiltrate the tumours, recognize the cancer cells, and can ultimately lead to destruction of the cancer cells. And there are various phases in this immunity cycle, and we can use different combination strategies depending on where we are in terms of the tumour biology. Cold or immune desert tumours will require the combination therapy to basically lead to increased priming and activation of the tumour cells. Whereas tumours with excluded infiltrates, where the immune cells are already there, they just can't get into the tumour, may require different combinations that enable the immune cells to better penetrate into the tumour. Inflamed cancers, and this for lung cancer may be the group of 50% or higher PD-L1 expression, these tumours may and seem to be responding very well to single-agent immune checkpoint inhibitor therapy, but again, there's still room for improvement, and we can still look at combinations to possibly improve the outcome of this subgroup as well. Now, if you look at patients in first-line metastatic and first-line advanced non-small cell lung cancer, there's one interesting study, KEYNOTE-24, that was presented at the ESMO meeting in 2016 in Copenhagen. This is a randomized trial comparing standard platinum-based chemotherapy with immune checkpoint inhibitor pembrolizumab in first-line treatment for patients with non-small cell lung cancer. But the trial only allowed patients with 50% or higher PD-L1 expression. And as you can see, there was a substantially higher response rate of 45% for pembrolizumab compared to chemotherapy with 28%. There were even a handful of patients who had complete response to immune checkpoint inhibitor therapy. If you look at progression-free survival on the left side, again, early separation of the curves in favor of the immune checkpoint inhibitor, with 10.3 months on median compared to 6 months median progression-free survival with chemotherapy. And also in terms of overall survival, these early data show that there's a clear benefit in overall survival with a hazard ratio of 0.6. So if you select a group of patients that have inflamed tumours characterized by high PD-L1 expression, single-agent immune therapy can have a substantial response and has performed better than standard combination chemotherapy as first-line treatment for non-small cell lung cancer. Again, if you look at the other subgroups, because not every patient, this is only a third of the patients who have high PD-L1 expression, not everyone will be a candidate for single-agent immune checkpoint inhibitor therapy. And most of the combinations at the moment are focusing either on combination of CTLA-4 and PD-1 axis, or chemotherapy in terms of what has been going on in phase 3 studies. There's a whole range of other early phase trials that are exploring different combinations that tackle at different points in this immunity cycle, and we will learn more about these results in the next years to come. But as it stands, the randomized trials at the moment are with CTLA-4 combinations or chemotherapy combinations. Now, what is the rationale for combining chemotherapy with immune therapy? There are two main things we want to achieve. The first thing is, as we established earlier, there is some link between PD-L1 expression and response rate. And what we're trying to achieve with combinations is make patients who have PD-L1 positive tumours respond better, increase the response rate in those PD-L1 positive patients. But also the other hope is to have more patients with PD-L1 negative tumours respond better and ultimately increase the overall efficacy of immune therapy in patients by combining it with a second agent, in this case chemotherapy. Now, for many years we thought combinations of chemotherapy and immune therapy are counterintuitive because people were claiming there are negative immune effects of chemotherapy. But actually what we've learned is there's wide potential for synergistic activity between chemotherapy and immune therapy. For example, chemotherapy can reduce the T regulatory cell activity within tumours. Also, chemotherapy can help with cross-presentation of tumour antigens, especially if chemotherapy can lead to substantial cell kill. And chemotherapy can also, in a very dynamic way, change tumour cell expression and infiltration of CD8 cells and tumour cell expression of PD-L1. So there's evidence from many preclinical studies of synergistic activity between chemotherapy and PD-L1 treatment. What clinical data we have at the moment, there are a number of small cohort studies with nivolumab, pembrolizumab and atezolizumab, which all show impressive early phase activity in terms of response rates. Response rate, objective response rate are in the high 40s, 50s, in some studies up to 70% of combinations with platinum-based chemotherapy mainly and immune checkpoint inhibitors. And this led to a series of phase 3 studies which are ongoing, which investigate the benefit of adding immune checkpoint inhibitors to standard platinum-based chemotherapy in the first-line setting. One of these studies showed early results, and the Keynote 21 trial was comparing patients with carboplatin pemetrexed chemotherapy with and without pembrolizumab. And then patients were continued with pembrolizumab or could receive pembrolizumab at disease progression. This is not a phase 3 trial, this is a small randomized phase 2 trial with a total of 123 patients. All patients were chemo naive, and patients were not allowed to have EGFR mutations or ALK translocations. As you can see from the results, the response rates were substantial and were better for the combination of chemotherapy and pembrolizumab compared to chemotherapy alone. 55% with the combination achieved an objective response compared to 29% with chemotherapy alone, which compares well with what we know from other phase 3 trials. If you look at the expression of PD-L1 and response to combination, there isn't a clear group that does substantially better, apart from the group of 50% or higher PD-L1 expression, but we seem to have similar response rate in very small numbers for patients with PD-L1 negative tumors or patients with low PD-L1 expression if immune therapy is combined with chemotherapy, whereas patients with absent PD-L1 expression seem to be having less of an effect of chemotherapy when given alone. If you look at progression-free survival in this small randomized phase 2 trial, again there's a clear trend, there's clear improvement with the combination of chemotherapy and pembrolizumab for 13 months compared to 8.9 months with chemotherapy alone. However, the overall survival data at this point, as you can see on the left side of the slide, show no difference between chemotherapy alone and pembrolizumab plus chemotherapy. Now, if you look at the number of events, the study is not mature yet for overall survival, and we need to wait and see whether those results may change over time. But it's obviously a concern at this point in time whether the combinations of chemotherapy and immune therapy will lead to the same substantial benefits in terms of survival as we have noticed with single-agent immune therapy, and we need more time and results from other phase 3 trials that will be coming out in the next few months to confirm this further. If you look at the different subtypes of non-small cell lung cancer and the different possible immune therapy strategies, we have a subgroup of patients of around 15 to 30% who have high PD-L1 expression. High expression defined as more than 50% PD-L1 expression. And these, according to the data I shared with you today, are excellent candidates for single-agent therapy with PD-L1 or PD-1 targeting agents. What about patients with ALK rearrangement or patients with EGFR mutations? Again, for these patients, we need to wait for combination data of immune therapy and targeted therapy, and at the moment, we can't define the optimal standard for these patients. But for the large group of patients who are PD-L1 negative or have lower PD-L1 expression, although being PD-L1 positive, the question is, what is the best strategy for these patients? Should these patients be considered for single-agent immune therapy, for chemotherapy, for example, up front, or is there benefit of combinations of immune therapy, for example, with PD-1 and CTLA-4, or of combination of chemotherapy and immune therapy? Now, you know that there's a range of phase 3 studies ongoing, and there was a recent press release that a phase 3 study of durvalumab and tremelimumab did not meet its primary endpoint compared to standard chemotherapy in the first-line setting, but it was only reporting data on progression-free survival, and overall survival data are not yet available. So again, we can't definitively answer this question whether PD-L1 CTLA-4 combinations are inferior, equivalent, or superior to chemotherapy in the first-line setting. And we are eagerly awaiting results from phase 3 studies of chemotherapy, immune therapy combinations in first-line therapy, which should be awaited in the next few months. Where are we going with immune therapy in lung cancer? And this is really to finish the talk for today. We know for patients who have high immune or have inflamed tumors with high expression of PD-1 or PD-L1, we can consider single-agent PD-1 and PD-L1 targeted agents. But the problem still remains what we're doing with cold tumors, and there are different strategies that are being pursued. One is to bring T cells into the tumors, and this is with combinations, as we discussed, of chemotherapy, of CTLA-4, of other immune-activating antibodies. And there's a whole range of targeted treatments that are currently being explored in combination with immune checkpoint inhibitors to bring T cells into the tumors and therefore make the tumors more inflamed. Other strategies are looking at generating T cells and again then bringing those T cells obviously to the tumor, and there will be a lot to be expected from, for example, CAR T strategies, which are just going into the clinic in solid tumors as well. If we look at safety and management of side effects, I would like to share again a case report, and when we share case reports of patients, we usually focus on side effects. Having said this, most patients with immune checkpoint inhibitors, at least single-agent immune checkpoint inhibitors, will experience very, very few side effects compared to chemotherapy. Now, this is a patient we treated, 60-year-old former smoker who received first-line therapy with gemcitabine and cisplatin, and then relapsed within a year with lung metastasis, as you can see on the CT scan. The patient was started on an immune checkpoint inhibitor, and 4 weeks after we started the patient, he came back and was fatigued, reported on mild shortness of breath, had some diarrhea, and had also experienced fever. So it's a mix of different symptoms. Investigations show there was an elevation of liver enzymes, the platelets were lower, there were no signs of infection, and their respiratory function was normal. When we assumed that most of these symptoms are immune-mediated, the treatment was paused, the patient was started on systemic steroids, and as you can see, very, very rapidly the platelets recovered, the liver enzymes went back down, and patients and all the changes on the CT scan, which suggested pneumonitis, resolved very quickly, and the patient could successfully continue his treatment with the immune checkpoint inhibitor. Now, if you look at toxicity of immune checkpoint inhibitors, there is a balance between immune tolerance and unchecked immune response. And what we are essentially achieving with immune therapy is we are shifting the symptoms from immune tolerance more to an immune response. And sometimes we do this too well, and patients will develop immune-related adverse events, which are essentially a form of autoimmune reactions. And it can, in theory, affect any organ, but what we most commonly see with PD-1 or PD-1 targeting agents is inflammation of the skin, of the gastrointestinal tract with diarrhea, sometimes elevations of liver function tests, sometimes pneumonitis, endocrine effects, most commonly with hypothyroid, and very rare cardiovascular effects. General events like fatigue, fever, chills, infusion reactions are relatively rare. One key message for all immune-related adverse events is if you look at this slide and see that the reactions are down to a shift to the left, what we achieve with immune therapy, we are trying to shift this back to the right, and this can be done with steroids. So if patients on immune therapy develop adverse events, it is important to start them as early as possible on corticosteroids, because those steroids can very efficiently and very quickly counteract the side effects patients have. It's unclear at what time these side effects can occur, and some side effects tend to occur a bit earlier, but ultimately they can occur at any point in time, whether it's rash, diarrhea, endocrine effects, or liver toxicity. So just looking at the time point when toxicity occurs doesn't necessarily tell us whether it is